Matei wanted to know which specific mRNA codons encoded for which specific amino acids. In order to discover this, they took a tube and placed the homopolymer of their choice. In this example, we'll use poly-U mRNA and a cell-free extract system into a radioactive labeled amino acid and 19 unlabeled amino acids. They then incubated the samples in order for translation to take place. They did this with a different amino acid each time and eventually saw that phenylalanine was the only radioactive response to poly-U mRNA. This meant that UUU encoded for phenylalanine. They also did this with homopolymers poly-A, which coded for lysine, poly-C, which coded for proline, and for poly-G, which did not work for this experiment. Overall, they identified 3 mRNA codons that encoded for 3 specific amino acids. Nirenberg and Leder did an experiment called the triplet binding assay to identify codons. A trinucleotide, or mRNAs, as short as 3 nucleotides, were added to a mixture of ribosomes, 1 radioactive amino acid, and 20 charged tRNA molecules attached to certain amino acids. We use GUU trinucleotide in this example. They were then filtered through a nitrocellulose filter. This was to see which amino acids were bound. tRNA paired with ribosome-bound mRNAs stuck to the filter, whereas unbound tRNAs passed through. Amino acids that were not bound to tRNA passed through and were unreactive. After the filter paper was assayed, it showed to be radioactive with an amino acid left behind. From this, they knew that the trinucleotides, GUU in this example, which acted as what we call a codon in mRNA, coded for the complementary triplet sequence on the tRNA, which we call an anticodon. This would be our example of the tRNA anticodon, which coded for the amino acid that was left behind and showed to be radioactive after our experiment. Nirenberg and Leder discovered 61 codons with this method.